All right. Good morning, everyone. My name is Heather Forrester. I'm the extension specialist at the Hawaii Ant Lab. It's great to be able to talk to people from throughout the state, as well as on the US mainland and internationally. The little fire ants, just to kind of frame the issue that a lot of us might be familiar with, is that little fire ants are really a Pacific Island problem versus a US mainland problem. So at the Hawaii Ant Lab, we do a lot of, we lead a lot of research on this particular species. We also deal with the issue that most of the pesticides available to manage these ants are designed for other species. So we've been able to formulate gel bait, which I'll talk a little bit more about today, which people are probably familiar with, but there's a new ingredient that we can mix into it now. So at the Hawaii Ant Lab, we look to protect Hawaii from the introduction of invasive ants. One of the ways that we do this is through surveys at ports of entry. We also look to prevent the intra and intra-island spread. We have no authority on doing so, but we try to educate people on this tramp ant species. As the extension specialist, primarily what I do is provide practical treatment methods to a variety of stakeholders. And whenever possible, we do look to eradicate infestations of little fire ants. On the Big Island, we look to manage little fire ants because it's so widespread, but we work with other invasive species committees, the Department of Agriculture, and other organizations on other islands to lead eradication efforts. One last thing, the littlefireants.com is a great resource. If you haven't been there in a while, we have updated our website, so it's worth having a look at. We also post blogs about any up and coming information, some of which I'll be sharing today with you. Since many of you might already be familiar with the Ant Lab and what we do, I wanted to focus more on the 3 pillars and some updates that we have. So within the Ant Lab, we look at research, extension, as well as operations. Some research updates. Michelle Montgomery is the lead researcher at the Hawaii Ant Lab, and pictured here is Jack Fields. What they've been working on is developing a marking technique for little fire ants using rabbit serum. And this is a collaborative project with USDA ARS researchers in Arizona. The goal is to to mark ants with rabbit serums to then see how far they are traveling to forage for food. So many times we get questions, how far do little fire ants forage? And we really don't know. It can depend on a lot of different factors as far as how far they have to go to get food or resources. So this project is looking to help answer that question. Our research team has also been testing a preservative for the Hawaii Ant Lab gel bait. For those of you who are unfamiliar with the gel bait, what it is oil, water, xanthan gum, which is an emulsifier bringing together the oil and water. It also helps thicken this bait so it can stick into the trees. Peanut butter, and then our active gets added in. And the reason that this was developed was to apply into the trees because little fire ants can live their whole lives in the top of trees without coming down to the ground to forage on baits. Anyways, the reason I'm explaining that is so you understand the ingredients in the gel bait and understand that they can start to ferment and they are not shelf stable. So the Hawaii Ant Lab has been testing methylparaben as a preservative for the gel bait. And in the the first part of the research team has found that with that methylparaben, the bait has remained attractive for at least.
Hey there, you froze. Hello. Okay, you came back.
Some upcoming research that's planned includes some field efficacy trials comparing granular and gel baits with the active ingredient of spinosad. Currently, we have a couple of granular bait products with spinosad that we've had good results with. These would include Antix and Firefighter. The one downside to these two products is they're not OMRI approved, even though they have an organic active. The reason being is the carrier is a non-organic corn. Since I've been working at the Ant Lab almost 8 years now, there's been an outcry from our organic farmers. So this research hopes to give them a gel bait solution that they can apply in their orchards, for example. Another upcoming project we have is a collaboration with USDA Forest Service and the UC Riverside. The
Robert.
The parasitoid
Join
wasp,
the meeting.
Oracema, it has been detected on Big Island. There's more information on our website under news, but this parasitoid wasp is host specific and only attacks the big-headed ants and little fire ants. Currently, we do not know how widespread it is, and we're looking to do pan traps at different locations on Big Island to see the distribution, and are currently looking for funding. I know Michelle has been looking for locations on Big Island as well to set up these pan traps to try to detect the Oracema. So if anyone is interested, her contact information, michelle.montgomery@littlefireants.com is up on display. Some extension updates. By bridging the gap between our research and practical use, the pesticides are always changing. One of the roadblocks or hurdles that we run into is that a manufacturer may choose to discontinue or phase out a product, and then that means we have to kick our research back into gear, see what else we can get a special local needs for to replace old products to keep up with the demand. So some of you may be familiar with the product of Provant. This was an additive into the gel bait for ornamental plants. It is not for use on anything that's edible, but some people do want that option when it comes to controlling little fire ants. So the Provant manufacturer has phased that product out, but the Hawaii Ant Lab has gone ahead and gotten a special local needs for Advion WDG, which is almost exactly like Provant. It has the same active ingredient of indoxacarb, but the percent of active ingredient was different. So we had to go through a lot of trials to get the right concentration. Advion can be purchased in small packets or larger containers. One of the hurdles that we're facing right now is that it is not currently available at pesticide distributors in the state, but it can be purchased online. The Hawaii Ant Lab has been working with local distributors to try to help them to get that into stores and available to folks. So if you've used Provon in the past and would like to try out the Advion now, understand that Advion is a line of products, so you need to specifically get the Advion WDG. And then that can be mixed into the gel bait for application on ornamental trees. When it comes to operations, I wanted to focus on Oahu and what our Oahu team has been dealing with. Currently, there are 23 known sites on Oahu, and thanks to Spot the Ant Month, which happens every October, the Hawaii Ant Lab received over 100 samples from Oahu residents in a month and a half. And that's actually exceeds the number of samples that we received in the previous 11 months. So this shows the importance that it's really folks who are submitting these samples that are helping us to find new detections of little fire ants. And this applies not only for Oahu, but for Maui and Kauai as well. Looking back on Oahu, where there have been at least a full year of treatment, the monitoring of 5 sites has been done, and we're not finding any ants, so that's great news. 8 of the active sites are within 2 months of completion for their treatment cycle and will hopefully be entering the monitoring phase. So good news on that front. On a not so good note, the Hawaii Ant Lab did not receive any funding from the State Department of Agriculture this year. And it's not that they didn't want to fund us, they just had a shortage of funds themselves. So all of us at the Ant Lab are working on a 50% involuntary furlough. If any of you are interested in supporting the Senate Bill 2996 to try to get us funding to get us back on track, there's more information on our website. But please be patient with us if you do need help with anything, because we're only working at half capacity. So with that, just some contact information for each of our 3 offices, as well as our website again. And if we have time, I'm more than happy to take any questions that you might have.
So Heather, can you stay until Robin's talk? We will have question answer session after that talk.
Okay, sorry.
Okay, thank you.
Thank you.
Thank you, Heather. So our next presentation is a keynote presentation from Robin Tingitala. Robin?
Great, thank you.
Okay. So Robin Tingitala is Associate Professor and Ecologist, a Behavior Ecologist at Department of Biological Sciences, University of Denver. And her research mainly focused on to answer how animal behavior is shaped by evolution and response to climate change. And today she will be talking about that topic titled there. And it looks like the most of the research is done in the Hawaiian island. So it will be interesting to us. Robin, please.
Absolutely. Thanks so much for the introduction. It's really been fantastic to meet folks already today. My name is Robin Tingitala, and my lab at the University of Denver is really interested in understanding the roles of both ecology and behavior in rapid evolutionary change. And to do that, we studied 2 organisms, a cricket and a parasitoid fly, that have each been introduced here in Hawaii, one originally from Australia and one from North America. And today I'll share with you some really recent work that my lab group has been doing to learn how it is that the interactions and circumstances that these animals encounter in their introduced range have facilitated the rapid evolution of communication both within and between the species. So almost all organisms, of course, now live in environments that have been altered to some degree by human activities, whether that's through introduction to new locations, the spread of exotics, habitat loss and fragmentation, harvesting, pollutants, or climate change. And one consequence, of course, of environmental change is evolutionary change. For most of the last 150 years, we believed, though, that evolution was sort of a historical process. It was something that we thought took place in the past and over extremely long timescales. More recently, of course, it's become clear that evolution also happens on what we might call contemporary or ecological timescales, even in a matter of just a few months or years. And the underlying causes of that rapid evolutionary change seem to include things like the invasion of new habitats, the rise and fall of natural enemies, competitors, and resources, and frequently the anthropogenic influences that we talked about on that last slide. About 20 years ago now, David Resnick and Cameron Gallimbour set out to identify studies in which rapid evolution had been observed in natural populations. And I think that the principles that they identified in that study are still really important for our understanding. At that time, they were able to find 50 examples of rapid evolution that had happened in natural populations. And of those, all but 6 were associated with colonization. And they defined that really broadly, just to mean any situation where individuals from one population became established in a previously unoccupied habitat. So that included situations like human introduction and range expansion, as well as metapopulation dynamics, where populations are sort of subdivided in a heterogeneous environment. But why is rapid evolution so frequently associated with colonization? Well, it turns out that founding populations are typically really small. They consist of just a few individuals. They've gone through a population bottleneck, and they're subject to genetic drift. And after arriving in a new location, population founders tend to go through a period of really rapid population growth, during which they are subject to novel selection pressures, things that might not have been present in their source range. So my lab is particularly interested in understanding how the conditions that are conducive to rapid evolution impact animal communication systems. And we spend most of our time thinking about communication between the sexes. So our work is focused on sexual signals, things like frog calls and bird plumage that are typically used by males to attract females and to convince them to mate. And like lots of evolutionary biologists, one of the things that I find really intriguing about my traits of choice is that these sexual signals can be extremely variable, both among populations and species, as I'm showing you here, but also within populations and species. So what's responsible then for all of the variation that we observe in these sexual signals? Well, we think of signals as complex stimuli that integrate multiple sensory components, each of which is produced by some underlying morphology, physiology, and/or behavior of the signaler. So today we're going to conceptualize that here as the instrument. That instrument generates a signal that is broadcast out into the environment. And as a consequence of their central role to attract members of the opposite sex, these sexual signals are really conspicuous. And that conspicuousness makes them subject to many and varied selection pressures. It's probably fairly obvious to all of us that signals should be amenable to the preferences of choosers. But I also want to think a bit more about the sort of broader social and ecological contexts in which signals are being delivered. That ecological context can include things like competing signalers and transmission properties of the environment that vary from location to location. The conspicuousness of sexual signals also makes them subject to the often unwanted attention of unintended receivers. Things like predators and parasitoids that instead are using these signals to locate potential hosts and prey. How then do the processes that promote rapid evolution impact animal communication systems and specifically sexual signals? Well, during that colonization process, genetic drift could, of course, impact both male signals and female preferences for them. And individuals that found a new population possess a first sexual signal that may be very well or very poorly fit to the new environment in which they find themselves. That new environment includes, of course, some of those novel selection pressures that we've been talking about: natural enemies, competing signalers, and transmission properties of the environment that differ from those in the source population. For the last 15 years or so, I've been studying the evolution of sexual signaling in the Pacific field cricket, Teleogryllus oceanicus. This is a very broadly distributed cricket. It's native to northern and eastern coastal regions in Australia, but it's also island hopped through the Pacific to some really cool places like Fiji and Samoa and Tahiti. And sometime between 700 and 1600 years ago, it was introduced to the Hawaiian Islands as the crickets accompanied, we think, the first human arrivals to Hawaii. What's really important about that is that when they were introduced to Hawaii, they encountered a novel selective pressure that they don't find anywhere else in their range. The instrument that we'll talk about today that crickets use to produce their mating signals is their forewings. They rub the file, which is a modified wing vein with a number of evenly spaced out little teeth on it, against the scraper of the opposite wing, and you get bursts of sound with each wing closing motion. There are also a series of important resonating structures on the wings, things like the harp and the mirror, that project the sound that that stridulation process is producing. In this system then, the wing is the instrument, and it produces a signal. It's a long distance, loud calling song. I'll play that for you now. And that song then, of course, is subject to lots of different selective pressures, including intended receivers, female crickets, who are using these songs to locate males and to determine which males they want to mate with. In Hawaii, but only in Hawaii, the cricket also encounters a deadly eavesdropping parasitoid fly called Ormia ocracea. That fly was introduced to Hawaii from North America. And here, as in the southern US and Mexico, it is attracted to the songs that male field crickets produce to locate females. So the parasitoid will locate a singing male and sort of spray larvae on and around that male. Some of those will burrow into the body cavity of the cricket, where they'll spend the next 7 to 10 days literally eating the cricket from the inside out, before they finally dig their way out of the cricket, pupating and killing their host in the process. We also, of course, work on some of those environmental considerations that I mentioned earlier, things like social factors and transmission properties of the environment, but I won't have much time to talk about those things today. The overarching question that I do want to focus on then is, why and how has the ancestral signal generated by the wing instrument that I'm picturing here for you diversified over just the last 20 years, generating now this sort of rainbow of variation in cricket song in Hawaii? Over a relatively short period of time, my lab and that of Nathan Bailey and Dale Broder have found several new morphs of Teleogryllus that produce altogether different and attenuated songs. So I'll focus on a few of those today, the silent, purring, and rattling crickets that my lab has studied the most. And in case it isn't clear why that's important and why this is such a wild story, consider this. Both natural and sexual selection are important players in the process of diversification, including as sort of drivers of evolutionary change in these signals that are used in mating. Yet our understanding of the earliest stages of sexual signal divergence is really hampered by our inability to directly watch that process. It's difficult for us to picture, for instance, exactly how a new sexual signal might arise, because observing the evolution of a new animal signal is understandably incredibly rare. Instead, we're always looking back on that process, making comparisons between closely related species that differ already in their weird and wild displays. We're instead getting to watch that process directly and in real time. So if you know anything about our study system already, what you probably know is that in addition to typically singing crickets that I played the song of earlier, about 20 years ago, completely silent crickets also evolved independently on multiple different islands within Hawaii when they lost sound-producing structures on their wings. So that might be really problematic for a cricket. And yet the mutation that causes this silencing or flatwing morphology spread through one Hawaiian population from negligible to more than 95% of males in fewer than 20 generations between the late 1990s and 2003. So this is actually one of the fastest cases of evolution in a sexual signal that has ever been recorded. And why did it happen? Well, probably because these silent males are protected from that parasitoid fly. For about 15 years then, what I just told you was sort of the status of crazy rapid evolutionary changes in Hawaiian populations of Pacific field crickets. But in 2017, I went to Kalapapa National Historic Site on the island of Molokai. I was there to look for crickets because I was studying how anthropogenic noise impacts communication in singing insects. And I had learned that Kalapapa was a small, isolated, difficult to get to site, only accessible by this really beautiful hike down a cliff face or a rather expensive puddle jumping flight. So I thought that this might actually be a really great control site for some of my work, a place that was largely free from anthropogenic noise. But we didn't know if Pacific field crickets were on Molokai at all or not, and I wasn't particularly hopeful. So I went into Kalapapa for a single night, and I had with me a single Tupperware container just like this one to collect crickets. And after searching around for about an hour and not hearing a single calling cricket or seeing a single cricket, I was about to give up when we found a delightfully happy but very small population of our field cricket living right here on the lawn of the Bishop House. So I collected about 45 crickets in an hour there, and I crammed them all into my little Tupperware container. And I took them back to my room where I hoped that they would lay some eggs overnight so that I could take those eggs back to my lab with me. About a half hour later, though, I heard this cat purring in my room. And I thought, well, that's a little weird, but it wasn't that weird. There are often feral cats at our field sites in Hawaii. What was particularly weird about this cat, though, was that it was purring from the inside of my cricket container. That's right, they were crickets. The crickets on Molokai were producing a different sound that was reminiscent of a cat purring and that had little in common with typical crickets of the same species, except that we could detect that telltale temporal pattern of Teleogryllus song. So let me play what I heard for you. I'm first going to play that ancestral species-specific song for you so that you can get that stuck in your head. Now, this is what I heard and saw in my room in Kalapapa. So clearly, these two morphs of cricket sound different. And indeed, when we analyzed their songs formally, we found that purring crickets and typical cricket songs differed in all of the different characteristics that we looked at. Purring songs are much higher in frequency on average, they're much quieter, and they're more broadband than ancestral songs, which gives them that sort of noisy rather than tonal quality. But if purring is going to be a new signal, it needs to be detectable and usable for receivers. So we next asked if purring functions in reproduction at all. And the first thing that we did was to use phonotaxis or mate location trials, in which we released females in the center of an arena with one speaker broadcasting purring song and 3 speakers broadcasting nothing. And what we were interested in this experiment was which speaker the female encountered first. So we reasoned that if the purr was no better than a silent speaker at attracting a female, we would expect females to contact a purring speaker 25% of the time, but a silent speaker 75% of the time. So that null expectation is what I'm showing you in the gray bars here on this figure. What we found instead, though, was nearly the opposite. Females were far more likely to contact the purring speaker than a silent speaker, and this differed significantly from the expectation by chance. So in this experiment, overall, about 60% of females could hear and use this purring song to locate males. But remember that I started this talk by thinking about rapid evolution of sexual signals, and we don't know anything about when purring crickets arose or arrived on Molokai because we didn't work there prior to finding the purring crickets. So a really exciting development over the past 4 to 5 years of very regular regimented sampling is that we've identified purring males in some of our other long studied populations where we didn't find them as recently as 5 years ago. So purring truly is novel in these locations. We now have different ratios and combinations of ancestral, silent, and purring crickets in multiple populations, which makes this a really powerful study system to address questions about the evolution of novelty. So having found this cool new sexual signal in replicate populations, we set out with support from the National Science Foundation to track the evolution of this new signal in real time. And our first goal was to characterize what was happening right at the beginning of all of this, what we called time zero, as close to the origin of purring in some of these locations as possible. And given the conflicting natural and sexual selection pressures that have been historically important in this system, we're measuring song characteristics of purring crickets, as well as the wing morphology that generates the sound. We're also measuring the hearing ability of the crickets and the parasitoid flies. We're measuring preferences of crickets, and finally, preferences of flies. And the variation that we uncover here is that variation upon which selection could act to shape this novel sound into a more refined signal. Our plan then has been to reassess all of those traits every 2 cricket generations for the next 20 generations. So the goal being to track the very earliest stages of potential coevolution between male signals and female preferences, and between hosts and parasitoids. But as we embarked on that really fun work, one of my graduate students, Jay Gallagher, started to hear things in our data that he couldn't explain. Songs that didn't fit into one of these existing categories of ancestral, silent, and purring. But we hadn't yet seen any new morphologies that might be producing those new sounds. So we decided to take another approach here. We wanted the crickets to tell us what was going on. So we measured 9 song traits and 14 wing landmarks on a bunch of field-caught males from across the archipelago. And we used hierarchical clustering to identify clusters of male phenotypic variation, or what we might call morphs. The already really well-described silent male crickets don't produce recordable or audible songs, so they were excluded from this analysis. So when we clustered individuals based on song and wing similarities, we actually found 3 sound-producing morphs, so 3 distinct male types. We found the ancestral typical crickets that I played for you earlier. We found those purring crickets that we had already identified. And we also found yet another new morph that we call rattling. You'll notice on this figure that the rattling and purring crickets sort of clustered together, and that reflects the greater similarity in their songs. Both of these types of males produce what we're calling a relatively attenuated song. So I'm going to play that rattling song for you now. And we found in this study that there were some really kind of key characteristics that were important for distinguishing these different types of males from each other. So in this figure, we've actually mapped those below the dendrogram that I showed you on the last slide. So we're going to talk first about the instrument, the wing morphology that the animals are using to produce these sounds. We found that wing features were important in distinguishing purring crickets from the other two types, the ancestral and rattling. Males that clustered with the purring morph are missing some of the structures on their wings. So they're missing a mirror and they're missing or have a severely reduced harp, whereas the rattling and ancestral crickets have all of those resonating and stridulatory apparatus structures that we talked about earlier. We also found in this study that song characteristics allowed us to distinguish all three morphs from each other. Remember that ancestral males have those loud tonal songs with a really narrow range of peak frequencies centered right around 5 kilohertz. Both the rattling and purring males produce relatively attenuated songs. They're quieter, they're less tonal, more broadband songs than the ancestral one, with purring being the quietest and the most broadband overall. And if you take these two things together on the two sides of the slide, this is actually a really interesting pattern because we can't explain, based on this sort of landmarking morphometric approach that we took, how rattling males are producing a non-typical song. In fact, if you use principal components analyses to visualize relationships between composite wing traits on the x-axis here and composite song traits on the y-axis, despite these obvious differences in song, the wing morphology of rattling crickets is indistinguishable from the ancestral. So what else could be going on here? Well, we hypothesized that rattling males must have changes to something on the underside of the wing that we didn't capture with our morphometric analyses. One possibility here was the file, that vein that has all of the evenly spaced out teeth on it. That's a really essential component of the stridulatory apparatus and a structure that's not directly measured using landmarking of the wing. So we used some three-dimensional digital microscopy to inspect the file on the underside of field-collected ancestral rattling and purring male wings. And the results were so clear that we didn't even need statistics. Images of the files of every rattling cricket that we examined had these distinct sort of gaps in the teeth, but neither ancestral nor purring males did. So purring males and rattling males both produce relatively attenuated songs, but they do so in really different ways. And that leads us to the idea that what once was this sort of dichotomous silent or singing system is now one with the rainbow of morphs producing different songs that I alluded to earlier. So purring in blue has become quite prominent in 4 populations. Rattling in purple is quite prominent in another. Silent crickets in green are pretty rare overall now. And curly wing and small wing males in yellow and orange, which I didn't talk about today, are found at relatively low frequency in a few populations and also produce differently attenuated songs. Naturally, then, we next started to ask why this was all happening. What selective forces facilitated the evolution of these novel song types? We looked at both natural and sexual selection, and in both cases, we used phonotaxis experiments in the field to measure the selection happening both from crickets and from flies. So essentially, we captured natural mate location behaviors and host location behaviors by setting up field-based phonotaxis experiments in which we broadcast different types of song or a white noise negative control from speakers out in the field. And what we find for both crickets and flies is that positive phonotaxis depends on song type. So running along the x-axis here are the ancestral song, the purr, and that white noise negative control. And for crickets, we found that about 20% located purring song, which makes purring less attractive than ancestral song, but importantly, still better than white noise or nothing. For flies, we caught 47 female flies at traps that we set in the field that were broadcasting the ancestral song, while we only ever captured a single fly at a trap that was playing purring song, and absolutely none at white noise traps. And all of that suggests something really cool. It suggests that purring might be a private mode of communication detectable to crickets, but that protects them from parasitism. After discovering rattling crickets, we asked the same question about rattling song using similar methods, but only in the population where rattling is found. And the overarching message was exactly the same. About 40% of female crickets are phonotactic to rattling song, which is here in purple. And that makes rattling more attractive than white noise or purring, but less than ancestral song. And while all flies tested were attracted to ancestral song, none were attracted to rattling. So rattling also appears to be a private mode of communication. In summary, then, today I showed you that at least 3 novel morphs of the Pacific field cricket have evolved over the last 20 years here in Hawaii in the animal's introduced range. And that the 3 morphs, silent, purring, and rattling, each protect the cricket from a deadly natural enemy that was also introduced here, but they do so in different ways, either through the complete elimination of a sexual signal or through the use of novel sexual signals that are less conspicuous to deadly natural enemies, but still moderately attractive to conspecific mates. And finally, because this is a shorter talk, there's of course a ton that I didn't get to talk about today that we are actively working on. So I wanted to share just some brief hints about other aspects of this system that we're working on, including our continued tracking of the evolution of songs, wings, and receiver responses over time, looking at the evolutionary history and genetic basis of both purring and rattling, investigating how the sensory systems of intended and unintended receivers, so female crickets and female parasitoid flies, might shape the evolution of these novel signals. We're starting to look at the community consequences of novel signal evolution. So how does this impact other crickets that live here? We're looking at divergence of purring across the archipelago, and investigating the roles of both plasticity and relaxed selection in the origins of these new signals. And then finally, we have some work going on host location behavior and how that might differ for novel versus ancestral hosts. And with that, I just want to say thank you to current and former lab members and funding sources who made all of this possible, and especially to my co-authors on all of this work who are listed at the top of this slide. And again, I want to acknowledge Dale Broder, who is my partner in crime on all of this work and who some of you know. Thank you very, very much. And I am happy to take any questions.
Okay, cool. Thank you. Thank you, Robin. So I have a question. So cricket has different morphs. Is it based on the wing morphology or the sound they produce?
Both. So when we define something as a new morph, we're defining that based on a combination of wing and song characteristics. So we're using that sort of hierarchical clustering method to figure out where the natural breaks are in the variation in song and the variation in wings that we see. And it's the wing morphology that's producing the song. So when you have a change in song, that's sort of underlain by the change in wing morphology.
So the purring sound or whatever sound they produce is based on the wing structure or morphs.
Yeah,
If the
yeah.
same
So
wing, can the same wing produce 2 kinds of sound?
No.
Join the meeting.
To some extent, there is some plasticity. So if you were to take, let's say, a rattling male and rear those rattling males under different environmental conditions, you might find slight variation in their song. So for instance, if they are reared in a drier environment, you might get some changes in how stiff the wings are that would change characteristics of the song. But they're still going to sound like a rattling male, which is dramatically different from a typical male, which is dramatically different from a purring male. So you can't switch morphs within an individual, but you can find small amounts of plasticity within a morph.
So that means we can say there is an evolution in wing morphology, right?
Yes, absolutely.
So there is a 3 morphs of cricket
At
here
least.
on Kauai or in Molokai. There is, I
Yep.
think there is a question on question by somebody. Is the crickets on Maui?
That's a great question. So I started working on these guys as a graduate student, and my advisor, Marlene Zook, had been working on them before I started in her lab. And she did in the early 90s surveys across all of the Hawaiian Islands when she initially found the crickets to see where they were. She never found any on Maui, but a question that I've had for a long time, and I haven't been back to look, a question that I've had for a long time is, what if Maui is actually the original source of silent crickets? So the way that she searched for crickets and the way that any cricket researcher would search for them is actually by sound. So you kind of drive around really, really slowly with the windows on your car down and hop out whenever you find sort of a field that looks like a reasonable place for the animals to live. But you would identify that the cricket was there by sound. So it's possible that these crickets are on Maui. We don't work on Maui just for historical reasons, that they were never found there originally. But it also doesn't make sense to me that they wouldn't be there, right, given the location within the islands.
Okay. So, okay, thank you. I have a question for Heather. So there is a Senate bill, and I think is that still on?
Sure. So we started off with a House bill and a Senate bill. The House bill was proposed by basically half of the House. It was a lot of people that had introduced the bill. And because there were these companion bills, the House bill fell off, but the Senate bill has crossed over into the House and was actually heard today in the Agriculture Committee. So then we just have one more round of hearings with Finance. And historically, that's where a lot of bills die, right? When it comes down to writing a check. So hopefully it works out. And
okay. So I have another question for Heather. So what does the wing? So I can see some wing ants in my house, in my kitchen or something. So what does that mean? So this is
Number one,
wing morph, yeah?
you-
I was talking about the morphs of insect. It's a wing morph. So what does that mean? They are coming into, they are making a, what do you call, nest in my house? Just curious.
So with ant species, they have the males and the queens will have wings. And some of the species will have a nuptial flight. And then once they've mated, the male will go off to die and the queen will shed her wings and start up a colony. Without looking at it under a microscope, though, you might want to have that checked out to make sure that it is, number one, an ant. But with little fire ants, I like to call them the penguins of ants because we don't observe them having that nuptial flight or flying to spread to new locations. So
the little fire ant doesn't fly. That means doesn't have a wing morph, right?
they have wings, but we do not observe them flying.
Okay.
Yeah. So
Okay.
for example, when we're using the gel bait with Tango, which is an insect growth regulator, that's going to slowly reduce the number of workers because the queen's not producing more. And then we see the winged queens coming out to try to forage for food. So that's a common time for little fire ants, that particular species, to come out and you would see the queens.
Okay, thank you.
Yes.
So another question. How gel bed differs from somebody's else? How many types of gel bed are there? So
the main reason that we developed the gel bait was to apply it into the trees and to use a product that we could get a special local needs permit for. With other homemade baits, if you're mixing your own, that's considered a reformulation of pesticides. So you could potentially get in trouble for that. There are ready-made gel baits such as taro, Terro, for example,
Okay.
which are more for a sugar-loving species. So with ants, it's kind of like going fishing. You want to use the right bait to catch the fish you want. Ants can be sugar-loving, protein-loving, or oil, fat-loving. And in the case of little fire ants, they're an oil-loving ant.
Oh, okay. Oil. Okay, thank you. So
Yeah.
another question for Robin. So cricket came from Australia, and the parasitic wasp came from somewhere else, not North America, somewhere from the. How do they associate here on Hawaiian Islands? Is that
So
yeah?
How did they?
Yes.
Right. Yeah. So we don't actually know when or how the parasitoid fly got here. It's a parasitoid of other field crickets in North America. And we know that the population here in Hawaii is most closely related to parasitoid flies from California. We have always thought that the fly must have gotten here at some point after Teleogryllus did, because native Hawaiian crickets actually don't sing in the right frequency range and aren't big enough to harbor the parasitoid larvae. So they must have been using an introduced cricket sort of the whole way through. But we haven't been able to kind of do the work to date their arrival here in Hawaii. So we don't know how long the two have co-occurred. The earliest, I think the earliest record of the fly is from the late 80s, I want to say. And like I said in the talk, the cricket has been here for somewhere between 600 and 1600 years.
Okay, thank you. So I will go. I will look at the chat box now. So there was lots of questions. Maybe could not hear purring. Maybe okay. If we have time, we will hear purring sound again. So
can I say one thing?
Yes, please.
I think that it's interesting that we presented together, because in your case, the parasitoid is bad. And in our case, we're looking as the parasitoid is the good guy here. So
right, yeah.
So cricket communicate. So everybody all the organisms communicate. In case of insect, I think they have some kind of other cues, visual cues, olfactory cues, and this one is kind of new. So what do you think about all those three kinds of cues in communication? How does this sound came in? Does all the insects, mostly in insect, they produce sound?
A lot of insects do, and actually more produce substrate-borne vibrations, so waves that are traveling through the ground or through the leaf litter or through the grass, than actually the kind of traditional, auditory traveling through the air kind of sound that we would think about. So yeah, for crickets and for insects in general, we absolutely think about all of those different modalities. Sometimes they are sort of combined into displays that they're doing. So you might have like a peacock spider that does, a dance that's showing off colors. It's also using movement, and it's using substrate-borne vibration through the tree that it's on. So they can all be sort of combined into a single display. For us, the crickets, because they're nocturnal, are not very visual. So most of what they do actually is after sunset. And in part, that's also because of this parasitoid fly. So the parasitoid fly is most active at dusk and at dawn. And so to avoid that, over time, the cricket has evolved sort of a later time of night that they really ramp up their singing and get going. So if you were to look at like other parts of, where the cricket lives in Australia, they actually will sing sometimes during the day. And here they really don't get going until after it's completely dark outside. So visual signals not as important for this particular animal, but they do use olfactory cues. They have cuticular hydrocarbons, kind of pheromones in their cuticle that they can use to determine the sex of individuals that they're interacting with. So they're smelling each other. They are using tactile interactions during courtship, but primarily they are using this sort of auditory channel. We are doing some work, and that's led by my collaborator, Dale, looking at how the songs that the animals are producing are traveling through, through the air as a substrate, but they also simultaneously produce substrate-borne vibration. And even silent crickets that we can't hear and that female crickets can't hear will produce that substrate-borne vibration. So it's possible that they're still receiving some cues in these other modalities, even when that traditional song is gone.
Thank you. So there is a question: how does the cricket hear? Does they have some kind of organ like tympanum?
Yeah. They do. They actually have they have ears in their legs, so in their kind of front elbows. So it's really cool when you're watching them behave. You can absolutely tell when they're doing what's called phonotaxis behavior, so moving toward a sound, because they use the sort of time that the sound arrives at each of their ears to determine which direction they should move to locate that sound. And that produces this really sort of telltale phonotactic response where they sort of wiggle in a line as they're moving toward a sound and trying to locate it. And luckily for us, their hearing is actually, the range of hearing, the frequencies that they hear best is relatively similar to what humans hear. So we're able to detect and use our own hearing in some capacities to determine what the animals can hear. We also are doing some neurophysiology experiments with folks who are really, really expert in, sticking kind of probes into the brains of these animals and figuring out which of the sounds that are being produced are actually being detected and register as a sound or as a song. Okay, there is another question asking, how do you pick up those sounds for yourself or whoever is recording or doing research? Yeah, we set up basically like on the go recording studios. So when we record these sounds, because the animals are, the purring crickets especially, are very quiet, as you noticed, you can't actually hear them when there's background noise of, a refrigerator or whatever running in your house. So we actually unplug everything, close ourselves into a small room, and we have a really standardized setup of digital recorders and microphones that we use, placing the animals at a set distance from the microphone so that we can look at things like how loud the song is, the temporal pattern, the frequency of that sound, how broadband it is. And I actually have one of my graduate students does all of our sound analysis. He's actually a professional musician and producer and came back to graduate school because of an interest in combining his music background with evolutionary biology. So he does a lot of our sort of nitty-gritty song analysis. Okay. So there is another question asking, is there any genetic work done on crickets to see the relativeness or the yeah, absolutely. Yeah, so Nathan Bailey's lab at the University of St. Andrews has done most of that in this system. I've done a little bit. So we have done population genetics and population genomics to try and understand the pattern of movement through the Pacific. Nathan was able to date the cricket's arrival here. He's also looked at gene flow between the Hawaiian Islands. So where are crickets moving to and from? Because we do have these morphs popping up in multiple locations and relatively, close proximity in terms of time. And then I did some work early on to identify that the mutation that causes crickets to be silent is on the X chromosome. So we know the location of that mutation now along the X chromosome, and we are currently working to kind of map the location of the purring mutation and also rattling to sort of compare that to this rapid evolution on the X chromosome that happened with the silent crickets. Thank you. So sure. lots of participants says it's a fascinating work. Thank you. Yeah. So I will give you a break. So Heather, what is the LFA status on Kauai?
There's a question.
Currently, there are 3 known infestations that are being handled by the Department of Agriculture and the Kauai Invasive Species Committee. The best way to get updates about what's going on your island is stoptheant.org, just to keep up to date on the HAPs. One thing to just throw out, since Kauai came up, I had the opportunity to work with Limahuli Gardens to come up with a prevention and early detection plan. So if there's any organizations out there that are in need of some type of plan like that, I'm more than happy to work with them. And I'll type my email in the chat.
Okay, thank you. Thank you, Heather. There is another question on methylparaben. Maybe you can answer in the chat box. I will move a little bit faster because
Sure. Sure. So with the methylparaben as a preservative, it would be an optional addition. I'm not sure the direction that the lab's going to take, if there's potentially an opportunity to share this information with the manufacturer to get something that you could just buy the gel bait. So because right now, if that's one of the main reasons, that one of the main hurdles for people is that mixing up this gel bait, though it is relatively easy, it's an added step. People are busy. So having something to buy ready off the shelf could be useful. So I'm not sure if that's the direction that it's going to go in, or if it would just be an optional addition. Currently, the gel bait, once mixed, is only good for 24 to 48 hours before it starts to ferment. And if the bait's not tasty, the ants don't eat it. So.
Okay. Thank you. So this question is for Robin. It looks like interesting question. So there is an evolutionary change in the cricket. Is there any on the flies as well?
Yeah, that's a great question, and one that we are very actively working on. One of the things that we think is really interesting is that even though I showed you those data from the field where the flies don't seem to be able to locate purring and rattling crickets from far away, if you bring them into close proximity, and we do this by just bringing them home with us essentially when we are working here, and sticking them into butterfly cages, you can play songs from short distances for them, and some proportion of the parasitoid flies can actually locate a purring cricket or a rattling cricket. But that's not usually the way that they hunt. So when they're out in the field in nature, they are hunting only by that acoustic sound. We think that they live in the trees, so they're actually coming down from really high up and need to be able to hear that sound from long distance to be able to locate a potential host. But this idea that they might actually change the way that they're hunting so that they are more likely to run into some of these novel cricket types is really intriguing to us. We have some preliminary data that shows, for instance, that when the flies are hunting for a typical song, they sort of beeline straight for that song. They have incredible ears. They're actually a model for hearing aids for humans because of the sort of teeny tiny distance at which they can, place the ears across their head, but still hear the same frequencies that we do essentially. And can, isolate a single sound from all of the background noise really easily and get around this sort of cocktail party problem. So they have just like phenomenal hearing ability. They beeline straight for an ancestral song. When we play a non-typical novel song, like a purring song, they actually use sort of a different behavior that looks like visual hunting might be important. So they'll move toward the sound and then sort of circle on their way down to that animal or the speaker that's broadcasting the song. And we know that these flies do use some sort of minimal visual hunting. That's why they're actually most active at dusk and at dawn. They need the sound of the cricket that happens at night, but they also need a little bit of light to sort of aim the larvae in the right direction and make sure that it lands on the host animal. So it looks like they might be potentially changing some of their host location behavior in a way that makes them more likely to encounter these novel types, or more likely to be able to successfully find a novel type.
Thank you. So next question is for Heather. So it's asking, what is the status of LFA in, looks like in the Mililani Mocha site?
Yeah, I was trying to text with my coworker on that one. I think he may have left the call already. Chris, oh, he's, so Chris has responded that he doesn't have audio, but the original Mililani Mauka site has not detected LFA in about 5 years, which is great. There is a smaller population nearby that is still undergoing treatment and will be assessed for ants at the end of April.
Okay, thank you. Thank you.
Thank you, Chris.
Also. Yeah. So there is another question for Robin. Do you think that flies in the trees are hunting tree crickets? So I don't know. There is a tree cricket is the same that you are studying?
Yeah, so we are studying field crickets, and in their native range, the flies only hunt field crickets, and it's because of sort of the structure of the body, the size of the body, and the structure of the wings is what dictates the frequency of the songs, and the fly's ears are really tuned to a very specific frequency. So they have co-evolved together with field crickets for a long time, which makes their hearing most pointed at that frequency that field cricket song is produced at. Tree crickets actually produce songs at different frequencies, but I would not, I think, like, had you asked me that 5 years ago, I probably would have said no, definitely not. After seeing flies locate some of these novel song types, I would not be so sure anymore. So I think it's possible. And one of the things that we are doing, actually in collaboration with some students at Kauai High School, is starting to look at the sort of community effects of these rapid evolutionary changes, right? So if we've got a sort of dominant host that has now either gone silent or is producing these really, really quiet calls that are basically private communication among crickets, what does that mean for the flies? Well, it means they have to find another host, right? So we have at least 3 other field and house crickets identified. All of them are introduced that we think are reasonable hosts for the fly now. So we have done tests where we ask the flies if they like their songs. We infest them artificially with parasitoid larvae and look to see if they can hold them all the way through to pupation, which is a problem for smaller things like tree crickets. They wouldn't be able to do that. It's a good sized parasitoid. It's like a house fly size. So we don't know, but we are expanding our sort of sampling to include basically every singing insect that we can find at our sites as potential hosts so that we can look into questions like that.
So another question, this will be a last question. Okay, so are you seeing reduction in the parasitoid fly population?
Not as much as you might expect, which is part of why we are looking at other potential hosts in the area. We did, when the crickets went silent, we definitely saw a reduction in parasitoid flies, but they never actually completely disappeared, which is curious, right? If they're using song only to hunt for potential hosts, if everybody goes silent at some locations like in Wailua at the ag station here on Kauai, we didn't hear a single singing cricket for about 7 years, and yet the parasitoids were still there. We know now that purring is increasing in frequency there, so it could be that they're locating purring hosts, but there are plenty of other things around that could also be involved in this interaction.
Okay, great, thank you. Presentation is Melissa Valdez from Oahu Army Natural Resource Program. She oversees most of the program in Oahu Army Natural Resource Program, and on Oahu ecosystem restoration and invasive plant species control. So today she will be talking about control of in the specific invasive tree species across managed native forested area on Oahu, and evaluating results and what next. Okay, Melissa.
Yeah.
Please.
Hi,
Yeah.
good morning. Oh, yeah, it's still morning time. I'm the only non-insect talk, so that's fun. So yeah, my name is Melissa Valdez. I'm the Vegetation Management Program Coordinator for the Army Natural Resources Program on Oahu, which is under the Office of Vice President for Research and Innovation, the Research Corporation of the University of Hawaii, funded by the U.S. Army. So I'm born and raised on Oahu, from Wahiawa. I've been with this program since 2017 as an intern and a student hire for years, and then on the field team for many years. And now in my current position, which I've been in for like the past year and a half, I get to oversee all the invasive plant control and ecosystem restoration. So yeah, I just thought that this would be a really interesting topic to present to a larger group with this weed control technique of targeting species that are relatively low density in our management areas. So these areas we fence off, we have our rare taxa that we manage around. And we've been trying to figure out ways to meet our native vegetation goals that we have laid out, and expanding weed control in a larger landscape rather than just focused around our rare plants. And over the years, these management units that we have these vegetation monitoring transects that go across the unit, so we get an idea of what the trend is, what the native or non-native canopy vegetation structure basically over years is changing. And so looking at our method overlapped with our vegetation results have given us these, I don't know, just an interesting way to like figure out what's the next step for how we need to change our management. So a little bit of a background, because I'm not too sure if a lot of people know about us. We are contractors from the U.S. Army. We manage 51 plant species, one endangered bird, the Oahu elapile, endangered tree snails, so specifically Acatella mustelina, and some other species like the picture wing flies, damselflies, yellow-faced bees, or bats. And our main goal is to stabilize these populations and control all threats. And that includes potential threats, like, for example, fire. So a lot of it is geared towards invasive plant control. But we do other things like slug, ungulate, rat, Jackson's chameleon, rosy wolf snail, you name it, we try to do it. It's a great program. If you ever want to work for us, you get to do a lot of different things. And we mainly work in the Waianae Mountain Range. We have a few areas in the Ko'olaus, but our majority of our work is in the Waianaes. So, like I said, in the past, our main focus in our management units were around just the rare resources, and these units are relatively large, and it's just really difficult to expand other than just those zones. And so what we were seeing with our vegetation and management unit monitoring results is that, of course, where we do a lot of management, weed control efforts, we see, you know, good positive results. We see more native recruitments and whatnot. But in the areas that we don't spend a lot of time, unfortunately, we just see this continued decline in vegetation, and it's becoming more non-native. So we tried to figure out, you know, what could we do to really get at outside of our rare plant zones. And it really, I guess, doing these targeted sweeps really took off when this incision point application method was introduced to us back in about like 2013 by James Leary. And it's a relatively lower effort as compared to if you were to go and, you know, cut stump or even a girdle method or whatnot. You basically, this picture is perfect. You would do like a little tiny notch and then apply a really small amount of pretty much 100% of a pesticide, herbicide into this tree, and you're able to cover a larger area in a smaller amount of time. So we really wanted to increase these types of targeted sweeps in our main management units, meaning we have a lot of managed taxonum. So Polychaea, we targeted Marillifia. In Kahanahaiki, was mainly Grevillea. And then Kalua'a and Maeli, Manuai, Makaha, we wanted to try to get at this Tuna ciliata, which is kind of, it's kind of scary because it's not there yet where it's monotypic like strawberry guava, but it has the potential to become pretty monotypic. And so we wanted to get at that before it became a huge problem. So what it kind of looked like, we didn't target every single tuna or every single Grevillea in those management units. We tried to identify the real huge trees that, in theory, if we got those huge mother trees, we would reduce the amount of recruitments throughout the management units. So what it looked like was identifying the huge trees, conduct the IPA, take a GPS point, and then maybe in about a year or two, depends how large of a tree, it's usually about a year or two, you'll see a complete defoliation almost. So really good success. Kahanahaiki was great, and it really worked for Grevillea. So back in 2015, after we did a bunch of vegetation monitoring, we identified a bunch of Grevillea throughout the management unit. So those are the red dots. And then we made it a priority to do those targeted sweeps. And by 2018, it dramatically reduced to 2 known locations of Grevillea, those yellow dots. Polychaea, so also a really good success story. So actually, Marillifia was controlled throughout this whole management unit. So this blue is the fence. And so they did a bunch of control throughout, but they wanted to see whether or not maybe the understory might have changed drastically because they removed such a large canopy. So they did this point intercept monitoring below the bases of some of those treated trees. And what they found is that they really didn't, it didn't really affect the way the vegetation changed with that. So it was great because then the canopy, we were able to remove it without really drastically disturbing the understory. But, and this could have been some other factors. It might have contributed to potential landslides because these trees are huge and a lot of them were found on ridges, and it may have, yeah, anyways, it may have led to some landslides. So what it kind of looked like, and this is a great a great picture to really show the amount of area you can cover. And these trees, in these red circles, those are all treated Marillifia. You know, they're crossing a few little ridges, a few gulches. This probably could be done in one day. So great way to just cover a larger area in a short amount of time. In Manawai, I just wanted to show like, so this left side, this left picture, this is an example of these polygon shapes. This is how we kind of track when we do weed control, we draw polygon shapes around the area. So normally we only work in these small little tiny polygon shapes, but IP or these targeted sweeps really allow us, and these are the blue highlighted polygon shapes on the right, it really allows us to cover an entire management unit. And then these little dots going across, those are the vegetation monitoring transects. So it's really great, like it's overlaps in a lot of areas, and we were able to track our success, I guess, and changes of vegetation. Same thing with Kalua'a, Wailea, and Makaha. I just wanted to show that you really can cover a lot of area, and we have those transects in place to track. And not surprising, from the results, the Grevilleo was really successful in decreasing their coverage using these targeted sweeps. But the tuna, we were still unsure. It didn't seem like we really saw that same response, and we were wondering why. And so we really have to think about, I guess, as far as what type of trees or invasive plants we need to target in these sweeps is probably based on their biology, the dispersal, seed longevity, growth weight that we don't have a lot of information about. So like thinking back at why was it successful for Marulaphia and Grevillea, it's probably because the seeds are probably getting dispersed a lot further out from the management unit or from that treated tree. So you're really seeing a decrease in that invasive plant coverage versus tuna. Unfortunately, you have, that thing sends thousands of seeds. A lot of it falls like directly below that mother tree. So when you take out that mother tree, you open up the light gap, and then the tuna, unfortunately, is like one of the fastest growing trees, just sprout up really quickly, and you're back to the same the same issue you had before that. Understanding more of exactly what the density and distribution is like in the management unit, we have to dive into that a little bit more, because in the past it was just based off of those vegetation monitoring transects, or just sometimes just casual observations walking through the management unit. Again, the vegetation management, the vegetation transects is nice because you do get kind of a trend throughout the management of what the coverage is doing, but it misses a lot of those possible pockets of tuna or Grevillea or any other type of target taxa. So it doesn't really give you a good sense of, or the entire picture of what the distribution is like. So, maybe we need to be looking more into aerial imagery, drones. Yeah, I just wish we had a lot of time to do those kind of things. And as a manager, we really don't, because things change so drastically in the environment. So I guess my goal is to present it to a larger group like this mini conference, is just to like hopefully have someone on this conference interested in looking at those types of characteristics that might make it successful for us to apply targeted sweeps, because we've seen success in a few. So yeah, that's all I have for today. Thank you for listening to the non-insect talk.
Yeah. Helen is also ecologist from, let me see. He's a research ecologist at the Pacific Island Ecosystem Research Center, USGS, in Hawaii Island. Her work mostly focuses on the distribution, abundance, and impacts of invasive species. And today she will be talking about distribution and host preference of Australian longhorn beetle on Hawaii Island. Helen, whenever you are ready.
Thanks, Roshan.
Mhm.
Yeah, and thanks everybody for listening in. I really appreciate being part of this sequence of talks. It's great to hear them. So yeah, as Roshan said, today I'm going to talk about the distribution and host preferences of Acalypta aesthetica, which is known as the Queensland longhorn beetle. So longhorn beetles are a diverse group with multiple problematic invaders. One of the best known of these is the Asian longhorn beetle, which threatens North American hardwoods and has been the target of an eradication campaign because the trees that it attacks are worth billions of dollars for forestry, recreation, backyard enjoyment. And the problem with longhorn beetles in general is that the eggs, larvae, and pupa tunnel in wood and can be easily transported in untreated wood. And so preventing longhorn beetle invasions is a major reason for regulatory standards on pallets and other solid wood packing material. So here in Hawaii, we have a relatively new invasive beetle, the Acalypta aesthetica. It's known as the Queensland longhorn beetle, or QLB. It's native to Australia and has never been invasive anywhere else globally, and so very little is known about it. The species was first detected in the state in the Puna area around Orchid Land in 2009, and then wasn't seen again for several years. But since then has emerged as a problematic pest. And so I want to start by acknowledging people from multiple institutions that have really led the work to date on this species, especially Stacy Chen at the Department of Agriculture, who was involved in getting the first specimens and understanding the host. Shenna and Scott Guide at the USDA, who've been involved in genetics and other analyses, trying to understand treatment methods. And Franny Brewer at BISC and Springer K as well, who are involved in public communication and outreach. And my colleagues, Bob Peck and Jordan Zarders at the Hawaii Cooperative Studies Unit. So sort of basics on longhorn beetles is that damage to host trees is done by larvae. So these bore into the wood and disrupt a tree's vascular system and even its structural integrity. And signs of infestation include sawdust-like frass coming out of the tree, as you see here in the picture on the right. And kukui also produce a lot of sap as well. And one of the challenges of managing these species is that the damage is all going on sort of deep inside the tree, where these beetles are basically out of reach from most insecticides and other treatment, even systemic treatment, because they're so deep in the wood. So as I mentioned, Stacy Chen at HDOA was the first to work on these species, and he's confirmed many of the hosts. So when an infestation in a new host species was reported to him, Stacy would bring the wood into his lab and rear out the individual to confirm that what emerged from that is indeed Acalypta aesthetica. So this is only a partial list of the species that he confirmed, but it includes many important host trees, culturally important species like kukui and ulu, agriculturally important species. Ulu again, cacao, citrus, avocado, common horticultural species like hibiscus and croton. And what's really striking about the hosts that Stacey's documented is just the incredible host breadth. So I put these species on a really simplified plant phylogeny. So here the species, if you look in the greens above, that they're not attacking, it's like mosses and ferns, right? And you can see that among the documented hosts are representatives of most of the major groups of woody plants. So conifers have a dotted line because it's infested cut pine logs, but not live trees. But again, it's this really striking host graph where we can't look at relatedness among plants to understand what this beetle is going to be a problem for. So given that, the group that I'm a part of was interested in a few questions. First, we know all these host species are attacked, but what tree species are preferred hosts? Are native species also at risk? So to date, the distribution of the beetle is mostly in lowland areas without many native tree species. And then finally, we were documenting the current distribution and evaluating methods for
join the meeting.
How to do that better. So this is work I've done with mostly with Bob Peck and Jordan Zarders. All right, so to understand host preferences, what we did is we reached out to private landowners and did surveys on invested properties. Every tree on the property, a known host or not, was searched for signs of infestation. And what we did is we analyzed host use given host availability. So the idea here is that when a property is infested, the beetle had a choice of which of the trees on the property to infest. And so by analyzing which of the ones it chose to attack, given what was available, we can understand its preferences. And this is an approach that's widely used in wildlife habitat selection, but I haven't seen as much in entomology. But here you can see the picture shows some signs of infestation at kind of the nook of one of the kukui branches. There's some sap coming out, and the other picture below shows some frass. And we did take samples of frass and of beetles, and Shenna did some genetic confirmation when there was new host species. So what that work found was that the favorite host of QLB is kukui, the state tree of Hawaii. So this figure shows the odds of infestation for other species relative to kukui. So kukui is that line at 1. And an odds ratio of 1 would mean that something is equally favored compared to kukui, and less than 1 means it's less preferred. So all the species are less preferred relative to kukui. And the second favorite host was cacao, which is about 1/3 as likely to be infested as kukui. And the other species were considerably less favored, and preferences among them were statistically indistinguishable. But again, these are all, what I want to emphasize here is these are all really important trees. Kukui obviously is the state tree, but cacao industry has been impacted economically by this beetle. It does cause death in both kukui and cacao and economic losses. So citrus and ulu and avocado, they're also species that are dear to us, and especially ulu, thinking about sort of sustainable agriculture across the Pacific, and then with citrus, this beetle is a risk to citrus industries on the mainland because of that. So I want to emphasize that these impacts on kukui, because that's where we've seen it kind of causing tree death the most. Again, kukui is an important canoe plant, which has a suite of traditional uses and current uses as a beautiful landscaping tree. Here I'm showing this tree on the left is a beautiful specimen used for shade and enjoyment. And on the right, a tree that has been heavily trimmed back after impact by this beetle. So it's definitely a concern. I do have one bit of good news from our surveys, which is that no infestations were observed on Ophia. And that's true even though the beetle is widespread in Puna, and so it's had plenty of opportunity to attack Ophia. So from there, I want to transition to what we're doing with these other native species. And again, that's because the field surveys were able to give us a view of preferences within the current distribution, but because those are mostly in lowland areas, the beetle basically hasn't met a lot of native plant species yet, really just Sophia so far. So we decided to do a lab rearing study to evaluate if native species are at risk. And thanks to Shanna and Scott for giving us some space to do this in their building. What we're doing is we're doing mostly oviposition testing. So females are collected from the wild and put in a bin with a branch of the focal plant species. And it's a no choice test. So the females only have one option. And what we're doing is we're asking, will they oviposit on this species if that's all there is? And so obviously there's caveats associated with that. Especially because cut logs can't mount a defense the way a living tree can. So for example, a living tree will try to sap a beetle out and push it out that way, which a cut log can't do. So this is kind of gives like a very baseline view of potential risk. So what we're doing is we're testing oviposition on native species, starting with the three I wrote here: Olapa, Ilex, the native holly, and koa. And after the female oviposits, we keep the larva in that same species and are basically asking if it can complete its life cycle. And this work is kind of preliminary results, but what we've found is that it looks like QLB can grow basically as well in Olapa and Ilex as they do on Kukui. So this figure shows a relationship between weight and head size, and it's similar across the different host species, whereas if a host was less suitable, we might expect a lower mass for a given head size. So this is very preliminary. It's kind of a slow work because the species develops so slowly. It can take 6 or even 9 months to complete its life cycle. And we've also expanded recently to doing some of this work starting with larvae that are coming out of Kukui. And that's because we've caught pretty few females recently. And so we're taking larvae out of Kukui and transferring them to track their growth. We're using ulu as a control so that they still go through a transfer between hosts and are comparing growth in koa right now mostly. And this mostly I want to show this picture because it's so striking. Jordan did this work and just the sheer number of larvae that can come out of a piece of infested kukui really highlights the damage that they're doing to these trees. So I think from a practical standpoint, this host testing of native species is really giving us a sense of which native species to monitor is our hope, and whether we can rule out impacts on any. Very anecdotally, it does seem like putting a beetle in with ohia, it hasn't done well. We want to try to get more adult female beetles to get a bigger sample size for that, but really kind of confirm in the lab what we've seen in the field, and then use what we are seeing in the lab to guide survey efforts in the field if the beetle does start to spread. So, we know that the beetle can complete its life cycle in Olapa, but we don't even know if the beetle can even survive at the higher elevation areas where Olapa is found. So there's still a lot of open questions here. And the last question I want to talk about is, the current distribution and how it can be quickly measured. So to cut to the chase, this is kind of a picture of the current distribution. What we did was, because kukui is a favorite host tree, Jordan's been driving roads and surveying kukui that are right along roads in publicly accessible areas. So this map shows just the results of those surveys, where orange is infested trees and blue uninfested. So currently the beetles found throughout Puna, into Hilo, and up Hamakua, at least as far as Papaiko and Pepekeo. But we haven't seen it in some kukui that are, in Hawaii Volcanoes National Park, and then farther to the west. And so while doing these surveys, we've kind of been thinking about methods for rapid assessment. So here's kind of a workflow for early detection and rapid response, which is, relevant across taxa. And so, something is found, it's identified and documented. And then there's this rapid assessment step where the goal is to understand the status of the invasion and basically inform planning. So it's figuring out what responses are feasible, what's realistic, basically looking across space and asking, how big is this problem? And so I kind of wanted to use the QLB surveys as like a model for thinking about how we do that better, still in a pretty low resource context, like in a pretty quick, rapid way. And so I was thinking about, what information do we have that we can bring together to guide surveys? One source is that Noah Lincoln and his colleagues map kukui based on remote sensing. So this isn't, of course, like every individual tree, but it's larger kukui groves. And on the right, I'm showing, because we're looking at this kind of rapid, publicly accessible survey, we're thinking about doing it on roadsides. So on the right, I basically imposed a 1 kilometer grid across the Big Island. And, the purple is where there's a road, but no kukui, according to Noah's map. And then the yellow is these roadside cells that had kukui in their grid. So the plan was basically to try to sample from these roadside cells and do it in a way that's weighted towards grids with kukui, towards places that are closer distance from known infestation, and then also places that are suitable for kukui. So, not going up to the top of Saddle Road and so on. And that's another layer that Noah produced. And we've been testing this out. It's actually been pretty challenging in terms of the feasibility. We're aiming to do like adaptive sampling so that when an infestation was detected, we would survey the nearby grids, like the grids all around it. And that's a sampling approach that's really good for rare and clustered species. But in many cases, we found that the grids nearby either don't have good access or don't have kukui along their roadside. So we don't get like a 0 or a 1, we get sort of like no survey. And also we would ideally want to map invasion intensity, so we can measure that by like the proportion of kukui that are infested. But most of these grid cells just have like 1 or 2 roadside kukui. So we've simplified, in this view to kind of a binary present or absent view of the invasion. But I'm still kind of very interested in kind of continuing to modify and assess how we can hone like a low resource approach to rapid assessment. So, as modelers, we often want the ideal survey, the ideal data, but in reality, there's, most species have few resources, and most species pose relatively low risk. And so there's this continuum of resource investment that's brought to bear to a given survey. And, on one side, there's sort of ad hoc approaches. Mostly there's not a formal survey strategy. And unfortunately, I think that kind of leads to overly optimistic view and imagining that, things can be eradicated because we don't understand the full extent of their invasions. And on the other side is, a formal, incident response team, the kind of thing that would happen with like a brown tree snake detection. But I think there's a real need for this, what I've highlighted in this like orange box here, for this kind of coarse rapid assessment. So how do we quickly estimate and monitor the invasion boundaries and the pattern of invasion within these boundaries? So that's something, kind of a broad question that my research program is interested in, and I'm still trying to use QLB as a model for that. Although I think in some ways it's actually easier probably with a plant where you don't need the host to be there to be able to do a survey. So mostly what I wanted y'all to take away from this is, QLB is a destructive generalist. Right now, kukui is hardest hit along with cacao. There's are not currently treatments available. So it's important, prevention is a really important strategy. Don't move wood or woody plants that might potentially be infested. The current distribution is in East Hawaii. If you're a property owner and interested in a survey, or a grove of kukui that we could access the survey, please do send an email. And I do think it's critically important to prevent inter-island spread, both within Hawaii and beyond. We don't yet know if this species impacts, the productivity of ulu, but I think it's really important from a perspective of sustainable agriculture throughout the Pacific to try to contain it here. So, and then finally, the rapid assessment methods are something that we need some more attention to. And something I hope to continue to work on. And then, so I'd like to mahalo everybody, some funding from USGS. Jordan Zarder's really led the field work along with Blake Mile and others, and everyone who's been involved from across the agencies. So with that, if any questions, I see one in the chat is, oh no, I'm not supposed to take questions now. I'll just be quiet. There's no lures.
Okay.
Yeah.
Okay, thank you, Helen. We will have question and answer session after the last presentation. Thank you, Helen. So our last presentation is from Dr. Cheng. Dr. Cheng, are you there?
Yes, I'm here.
Okay.
I'm ready to go.
Okay, so Dr. Cheng is the associate specialist in turf grass and landscape management. So much of his work is focused on coconut rhinoceros beetle and lobate lax scale and other turf grass insects and diseases. But today he will be talking about hala scale, update on hala scale management research and its statewide distribution in Hawaii. Dr. Cheng, whenever you are ready.
Okay, great. Let me go ahead and share my screen. All right, good morning, everybody. Well, if you're in Hawaii, it's still morning time, and I realize I'm the last to talk this morning. So I'm the only person between you and your lunch. I guess I will try to keep it brief. So today we'll be talking about hala scale on our research updates and also its latest statewide survey that we did last year. My name is Zhiqiang Cheng. Most people just call me Cheng, much easier than my first name. And here is Mason is my PhD student, and Philip was my former postdoc. So they contributed significantly to this work that I'm presenting today. So first of all, I would acknowledge all my current and former lab members, graduate students, postdocs, technicians, and visiting scholars, and especially Mason and Philip, so that you guys can see how they look like. They are actually they handled this project pretty much for me. So for hala scale, and for folks in Hawaii, you guys are probably very familiar with it already. It's not a brand new invasive pest per se. It has been here since 1985-ish. I will have a slide on that later, but just briefly, we will go through the introduction very quickly because I think most of you guys know about Hala scale to some extent already. So it is part of the palm scale family, and also it pretty much attacks Halas. That's why it got the common name Hala scale. So we likely got it from Java and Singapore, but that's, we typically, that's kind of common for Hawaii. We got a lot of pests, pathogens, everything from tropical Asia because of all the traffics and also the location where we are. So we got a lot of those pests from there. And the Hala scales, like similar to many scales, mealybugs, and other those kind of insects, they are at the crawler stage. They can move around very well. That's the stage where they can spread quickly and easily. But when they become adults, then they fix themselves on a certain spot. They don't move around anymore. And the severe infestation can actually kill plants. But if you have a certain, mild or moderate, intermediate infestation on Halas, you will see the Halas overall is not healthy looking, tilted yellowish coloring. From the distance, you can see already that the tree is not doing well. And then upon close-up inspection, you will see those Hala scales on the leaves, both, or if the infestation is very severe, it can be on both sides of the leaves and also on the seeds as well. So why we are concerned with Hala scale? Because for two reasons, at least in my opinion. So first of all, like Rashan mentioned, my job here is turf grass and landscape pest management. Halas, they are fairly common landscape trees or ornamental trees in Hawaii Islands. And also they have Hala trees, they have very important culture values because mainly due to the weaving practice that people here do. So without a healthy leaves, if the leaves are infected with Hala scales, then the leaves are no more usable for weaving practices. That's why, at least from these two reasons, landscape values and the weaving culture values, Hala scales are, important pests that we want to and we need to manage within Hawaii. So first, so like I mentioned before, the Hala scales are not really new to Hawaii, and it was first confirmed a long time ago on Maui. But right now, everybody knows that it is widespread on Maui, and we have limited infestation on Oahu confirmed at different locations, 2013, 2019, and 2020 most recently, and also on Molokai. That was the first detection on Molokai was 2014, but right now it is widespread. We will talk about that later when we talk about the statewide survey result. And they were limited, but eradicated infestation on Hawaii Island and Lanai. For Lanai, the eradication was successfully done back in 2019. I'm not entirely sure about the Hawaii Island situation, but maybe folks from Big Island, I kind of see like Stacy Train is on the line, so he might provide details information on Hawaii Island, but I'm not entirely sure about that. But anyways, here, this is one of the three hotspots on Oahu where we have Hala scale infestation. This is at Chaminade University campus. So I was approached, Department Ag people reached out to me back in 2016, I believe. It was actually Derek. At that time, he was setting up another independent trial on his own. He was testing dinotephrine, and he approached me and see whether I'm interested in doing additional, trials on Chaminade University campus, where they have about 15-ish Hala trees all infested with Hala scale, but at different infestation levels. So with very limited number of Hala trees over there, and we were not able to do a proper replicated research study on those Hala scales control, but we did more like, I call this more like a demonstration project. So we tested very roughly, we tested imidacloprid via injection, imidacloprid via soil drench, and acephate via injection. So based on our research, well, it's a more like demonstration, like I mentioned. So either both the injection and the drench, they were fairly effective. When we use imidacloprid, we had about 70% mortality over 6 months of post-treatment. So that was a reasonably good control capacity efficacy. And at the same time, like I said, Derek from Department Ag, he was doing his separate study, and the dinotephrine was working very well as well. So coming fast forward a couple years, and I got some funding to actually design and work on proper, replicated research project on HALA scales control. And if you kind of recall more or less that HALA scale is only at several limited spots on Oahu, and I simply do not want to set up a trial at the location where we do not have active HALA scale infestation. So the ideal location to actually run this relatively large, field experiment is actually on Maui, because on Maui it is widespread. There's no concern wherever you set up the treatment. So when we ended up setting up the research project at CTAR's Maui extension office area, they have like a plot area where we can set up plants and do research experiments over there. So this was a 2-year project. We did the experiment in the first year, and we repeat that in the second year. So we essentially inoculated HALA scales on those potted halas, and they were very small to begin with, the halas, and then they grow fairly fast. So if you see the picture, they're much bigger than the picture I'm showing on this slide. So then for the evaluation, we designed this rating scale. So the rating scale goes from 1 to 5. This way, I call it HALA scale infestation level, or sometimes we can loosely call it a plant health rating. So it is visually evaluated by 2 people always by at least 2 people at the same time, because we want to avoid the bias from one person doing that. So the 1 to 5 scale is 1 to 5. 1 means no scale at all, but 5 means plant dead due to the extremely high severe infestation, and the 2, 3, 4 are in between. We didn't really have the 5 situation, and we also didn't have the no scale infestation, because we did inoculate the scales on all those potted halas. So why we're doing this? It is becoming kind of challenging a little bit, because for the Oahu work we did at Chaminade University campus before, it was on Oahu, and we actually go there after we treated the halas, and we go there at a certain time, and we actually bring back the leaves. We do the visual rating, and also we bring back the infected leaves, and we actually set them under the microscope to see whether the scales were dead or not. However, we did not have that capacity to do it on Maui, simply because we usually go there in the morning and come back in the afternoon, and there's no very reliable microscopic lab for us to actually do the diagnostic over there. Plus, it's really time consuming, as we have a lot more. We have totally about 60 HALA plants, so it's not it's very hard to handle within a day. So we come up with an alternative way to do it, and we actually tested it several times in the beginning of the year 1 project. It worked fairly well. So what we did is we simply use a tape to tap on the leaves of the HALA scale, of the leaves of the HALA, and if the assumption is if the HALA scales are dead, they will not hold on to the leaves effectively, and with the tape you can easily peel them off. But with very healthy and active HALA scales, they will hold on to the leaves, and you will not be able to peel them off easily with the tape. The reason we are doing this is even we kill the HALA scales with all those treatments, they are still attached. The dead halas, their HALA scales are still attached to the leaves. That will affect our visual evaluation that I just mentioned before. So we did this several times to make sure this system kind of simple system works, and it did work. So we kind of included this secondary as additional measurement when we evaluated the treatment effects. So for this research project, we had a range of treatments. Some are relatively new chemicals, some are not so new chemicals, some are still chemicals on the horizon, not even labeled for scale insects. Some are not registered in Hawaii, but we want to see what options we have, just in case if they are in the market and registered in Hawaii for people to use. So we don't have to go through all these details of the treatments, but the experimental design, we had 5 replications for all these 11 treatments, and we applied the treatment based on the labels and also following the frequency of applications. So we had an automatic irrigation setup for the experiment. So we had a pre-designed scheduling of going back to Maui. It was during the pandemic time, so it's kind of difficult, especially in the beginning, that we have to do all those exceptions for travels. And my graduate student, my former postdoc, they traveled a lot just to go to Maui for this work. lot of paperwork, a lot of negative test result you have to show in order to actually fly from Oahu to Maui to do this work. But we did that. And the scale, we did the visual rating, as I mentioned. Also, we did the scale percentage of the scale removed by the tap removal method I just mentioned. And also we measured the plant height, plant canopy, and everything from the beginning, in the beginning, and also at the end of the study for within each year. Also, we did not observe any phototoxicity from all those treatments among the 2 years, within those 2 years. So I have a lot of results, but I will just show 2 slides as the key results in terms of actual control. So as you can see, we have different sampling, measurement time, but the graph I show you here is the last sampling collection measurement time within each of the trial years. So here is basically a lot of bars and everything, the analysis. But the quick point I want to mention is these 2 insecticides, Altus and Talus. Those are always jumping out as the best treatment. Basically from the 2 years research. So from here, plant health or HALA scale infestation level is basically the same thing. That is 1 to 5 scale. 1 is actually better. 5 is the worst. So then basically we are looking at the lower bars. The lower bars, the better in terms of a control capacity. So here you have these 2 at the resulting the significantly lower, significantly lower infestation level after treatment compared to the control and other treatments. And this is the first year. The second year data, we have these 2 treatments showing the lowest infestation level at the end of the season. There is another one, kind of become showing very good effects, but this was not one of the bad one. It was one of the relatively better ones, but not as good as these 2 chemicals that we identified in the first year. And similarly, when we use the tap removal method to represent the mortality of the HALA scales, and we get a fairly similar result. So basically for this removal is the idea is the more you can remove by tapping it, especially the more dead HALA scales are from that area you tapped. So that was the consideration. So in this case, we see in the first year, we have those 2 chemicals worked the best. So they removed 100% in this case. 100% means that basically means 100% of the HALA scales on that area where we removed randomly, located on each of the leaves, they were dead. So then that basically gives out this indication. And similarly, we have these 2 treatments showing very good efficacy in year 2 as well. So the key message here is these 2 chemicals, they were the best among the 11 treatments we tested among within this 2-year repeated research, replicated research. So statewide survey, that's coming over to, that's the research update, and then the statewide survey update. So at this point last year, before we started our statewide survey, we know this already. Maui already widespread, so there's no point going back to Maui just for the survey purposes. And Oahu and Molokai, we know that we have established infestation. Oahu, we know there are certain hotspots on Oahu, but Molokai, we believe it would have been widespread, but we want to double check that. Lanai and Hawaii, we know there were limited but eradicated infestation back in 2019. Hawaii, that was not reported from Hawaii. So this was basically we visited these 5 islands for this HALA scale survey work. So we mainly targeting plant nurseries where and a lot of garden centers and the high traffic areas with a lot of HALA trees as landscape trees where they are most likely to have infestation coming from other islands possibly. So those are the areas where we focused the most. So here is the Oahu survey sites. The yellow dots are the areas where we surveyed HALA for HALA scales, and the red dots are the positive locations. And these positive locations, I have a zoomed up map for that. So we already know that basically on Oahu, there were 3 hotspots for HALA scale. Waikiki area, several hotel and resorts, and the Chaminade University area, and also the lower campus, Dole Street on UH Manila campus. And we did this survey. We know that by 2020 already. And then last year, we did the Oahu island-wide survey, and we did not find any additional positive sites in addition to those 3 sites. So this Oahu remains these 3 hotspots. Molokai, as we suspected, widespread already. As you can see, all the red dots are positive sites for Hala scale. Lanai, in 2019, they eradicated the limited Hala scale infestation, and in last year, and we did not find any positive infestation on Lanai. And a similar story for Hawaii, for the Big Island. And for Kauai, good news for Kauai is there's no infestation recorded on Kauai last year. And we are hoping to repeat this survey later this year. My graduate student and we are looking for, applying for some small funding to actually do it. Here are some fun pictures. This is Mason, and fun pictures that he was, surveying all the islands for Hala scales, different locations, different, landscapes. He went for, to just to look for Hala scales. Okay, so quick summary. For the research part, we identified these 2 chemicals. They are very effective in terms of Hala scale control in our 2-year repeated replicated research project. And for the statewide survey, I just mentioned those, Molokai, widespread, Oahu, still the same spots. Lanai, Hawaii, and Kauai, there's no Hala scale detected as far as last year. And there was actually some movement or some effort on UH manure campus. Remember, the lower campus, Doe Street area is actually the 3rd hotspot of Hala scale infestation on Oahu, and that is part of the management under UH manure landscaping and ground maintenance. So then they reached out to us, and we reviewed the overall situation, and our recommendation is that either you treat those Hala trees, although the infestation level has been fairly high, treatment may not be very effective, or if they are willing, they could cut down the Halas. I wasn't sure whether they are willing to actually cut down those Hala trees, because those Hala trees are fully grown, beautiful Hala trees. But the director of the ground maintenance and landscaping there, she was actually very much willing to cut down those trees. She wanted to get rid of the Hala scale infestation from the campus. We sacrifice several infected trees, but in return we will keep the campus at least hopefully free of Hala scales for the next few years down the road. So that's happening actually right now. I believe they haven't cut down all the trees yet, but they are doing that right now. I think that's the most part of my talk. Always I have the disclaimer, because whenever I have chemicals mentioned, I have to have this disclaimer on that. And acknowledgement for funding for collaborators on Maui, especially Hannah and Norman. They were supporting us with maintenance, plan maintenance, and the data collection to some point. And also a lot of folks they helped during our statewide survey trips across the islands. And that's all my talk. And I will, let me see, I will have actually another very brief video to share with you. I had a project, I had a grant last year, and I hired a pest management association to actually produce some extension education materials for me. And I hired their video crew to come over here to Hawaii, and we shot a lot of videos. And during the process, they also produced 2 invasive species videos that I can make it to public, so that people can become more aware of it. One is coconut rhinoceros beetle. I'm not showing that here, but the other one is actually Hala scale. So it's a little bit less than 4 minutes. So I'm gonna try to play that from my computer and see whether we can do that effectively. So let's see. I believe I will have to unshare first and share a different screen. Yeah. So stop sharing, and let me go play the video.
Hawaii's natural beauty.
Okay, let me get that ready. Share screen with that only, and I believe there is a way to share.
We can see it.
You can see it, but I don't know whether you can actually hear it.
It's not playing though.
Yeah, I want to get it ready and play it. Yeah, I don't know whether you can share the voice as well. Advanced sharing options. Okay, let me play and see whether you guys hear
Is
it.
legendary,
Yes,
and
can
this
hear
chain
you.
of Pacific Islands is on the list of places
perfect.
to
Roshan, can you hear?
Yes.
You can hear? Okay, cool.
people. Blue skies, sun-filled beaches, and stunning mountains, along with the inviting charms of friendly people, only make that allure grow deeper. Another of Hawaii's most enduring qualities is its lush green vegetation. Hibiscus, plumeria, and several varieties of ferns can be found here in abundance, as can the hala tree, another of Hawaii's important native plants. Hala plays an integral part in preventing coastal erosion, as well as inhibiting invasive plants such as red mangrove. Its ability to thrive in salty and sandy soils has helped build its prominence in native Hawaiian culture. The hala leaves, lumber, and oils are all put to use by people for a multitude of purposes, such as landscaping, building, and traditional weaving techniques. Healthy plants have a vibrant green color with full leaf fronds and a robust, firm feel. But hala currently finds itself under attack by a very small and very powerful enemy, aptly named hala scale. Scouting for hala scale is relatively simple. These scale insects are most often found on the underside of leaves and appear as a small black dot surrounded by a white rim. Other signs include stunted leaf growth, reduced leaf blade size, and even decaying or dead leaf edges. First detected on the island of Maui in 1995, hala scale is now established on Maui, Molokai, and Oahu. A member of the palm scale family, hala scale adult females are encased with a shell-like exoskeleton. Offspring scatter from the female while in the corolla stage, the first stage of development. They can be spread by moving infested plants or leaves, or even carried on the wind or by birds or other animals. Once crawlers begin feeding, they attach by using their piercing sucking mouthparts and remain in place, feeding on the plant and slowly draining it of color, essential nutrients, and life itself. Though they are very small, only about 1/3 to 1/2 of a millimeter in size, roughly half the size of the head of a pin. Careful scouting can reveal them, most readily on the underside of hala leaves and in the crevices of seeds and plants. While biological control of this pest would be the most sustainable and effective means of managing an outbreak, no natural enemies have yet been found. Ongoing chemical trials, like the one shown here, conducted by Dr. Chung's lab at University of Hawaii at Manoa, have shown positive results with some products in combination with ultra-pure oil foliar sprays. These have been helpful in keeping infestations low and slowing the spread of hala scale. You can help too. Please do not move hala plants, seeds, or leaves between islands. Try not to visit other areas with hala after being exposed to an infested plant. And monitor hala plants at your location regularly for signs of leaf discoloration, or check the undersides for signs of the pest. By working towards solutions from wherever we are, together, everyone can make a difference.
Okay.
Okay, that was
great.